Welcome, I'm Yousif Fadhel, and I will be presenting my analysis of a newly conducted experiment on the potential use of telomerase therapy as a treatment for the HGPS disease. You may be wondering, what is HGPS? Well, Hutchinson-Gilford progeria syndrome is an extremely rare progressive genetic disorder that causes children to age rapidly, starting in their first 2 years of life. Patients may appear normal at birth, but signs and symptoms such as stunted growth and hair loss become prevalent soon after. The eventual cause of death of these patients is usually related to vascular issues such as heart problems or stroke. So, what causes HGPS? Well, a single letter misspelling in a gene on chromosome 1 called the lamina gene, which is responsible for coding the protein lamin A, causes HGPS. From our course, we know this type of mutation only affects one nucleotide in a gene sequence, and is also referred to as a point mutation or a missense mutation. This type of mutation produces a smaller morphed form of lamin A, which is called progerin. Lamin A has a primary and secondary protein structure and is an essential supporting component of the nuclear envelope. We have also learned in our biochemistry unit that the nuclear envelope is a double membrane made up of a phospholipid bilayer that surrounds the nucleus. Its job is to separate processes occurring in the nucleus, like mRNA transcription, from processes occurring in the cytosol, such as translation. Later in our molecular genetics unit, we learned that these two particular processes are the foundation of protein synthesis, so it is really critical that the production of lamin A is unaffected. In HGPS, it is also really important to understand that progerin remains permanently, and it will accumulate in the nuclear envelope, which will make cells unstable and alter their cellular composition. The article I chose is titled "Telomerase Therapy Reverses Vascular Senescence and Extends Lifespan in Progeria Mice," and it was conducted by the University of Edinburgh Medical School. It was published as recently as August 2021, and it contains many intriguing experiments related to HGPS. I particularly chose this article because it reminded me of a conversation Mr. Greenberg had with the class about the potential use of telomerase or telomeres for immortality. And this is a very similar concept, as it is proven to extend the lifespan in progeria mice. Now, the article title mentions something called telomerase therapy, which is a treatment created from an application of our body's telomeres and the enzyme telomerase. As we talked about in our genetics unit, telomeres are DNA protein structures that form protective caps at the ends of eukaryotic chromosomes. Their job is to protect our genetic information by preventing the loss of adenine, thymine, and guanine cytosine base pairs, which, as we know, are connected by hydrogen bonds. And what's really interesting about these telomeres is that each time a cell divides, the telomeres get shorter and shorter. When they get too short, the cells become what we call senescent, and they will undergo apoptosis, which is programmed cell death. This process is directly correlated to aging, as you can see in the GIF in the middle of the screen. So I made this GIF to show the aging process of a chromosome. As someone gets older and older, their telomeres get shorter until they no longer can function and become senescent, and that is when the cell will die off. Feel free to scan the QR code on the left for more information about how our telomeres protect our genetic information. Telomerase is an enzyme. As we know, enzymes are built from amino acids and tertiary and quaternary structures, and as we learn, they help speed up metabolic processes and chemical reactions. Telomerase specifically adds nucleotide sequences, which are the monomers of DNA, to telomeres. Now, what did the article present? Well, primarily, the experimenters identified that HGPS patients were experiencing drastic effects of vascular aging. They noted that vascular aging is characterized by an impairment of endothelial cells, and that the endothelium is crucial to maintaining vascular homeostasis. In class, we have recently learned that homeostasis is our body's natural bias towards a state of equilibrium. Vascular homeostasis, however, is just a little bit more specific, as it relates mainly to maintaining stability in our circulatory system through methods that we have discussed, such as vasoconstriction, vasodilation, and even the positive feedback loop of blood clotting. Endothelial cells are responsible for performing these tasks. These endothelial cells line blood vessels, as seen in the diagram of the artery here with the endothelium. In the article, the researchers hypothesized that lengthening the shortened telomeres of HGPS patients could potentially reverse the measures of vascular aging. They then presented two promising treatments. They transfected human endothelial cells with their treatment to prove that it was able to restore telomere length. And as you can see in the slide, there is a HGPS cell that was treated with telomerase therapy. and you then get a rejuvenated endothelial cell. So what is this treatment? Well, the two main treatments that the experimenters within the article assessed as a potential solution to help patients are two types of TRT modification therapy. The first one, labeled HTRT, is called human telomerase reverse transcriptase, and it adds new DNA segments to the ends of human chromosomes. The second one, MTRT, plays a very similar role to HTRT, but it is applied in mice. And the article's most fascinating discovery came from their experimentation on mice using MTRT. MTRT provided proof of viability for the application of telomerase therapy in a living organism. And through these trials, they observed that this telomere treatment had the ability to increase the lifespans of mice. Now let's take a deeper look into the experimentation conducted in the article. The researchers performed three main case studies. Firstly, to see how HTRT affects cell morphology and function of endothelial cells, then how HTRT affected the gene expression of the human genome, and finally the animal studies to see the effects of MTRT on HGPS mice. Next, let's take a look at gene expression. Transcriptional profiling by RNA sequencing showed that over 1,280 impaired genes in HGPS endothelial cells compared to the unaffected parental control. Application of the HTRT treatment found that over 265 of these genes were fully rescued by the therapy, and several others were partially saved. It was also revealed that HTRT treatment reversed various other HGPS-affected biological processes, most notably of which being DNA damage and DNA repair. Finally, let's take a look at the animal studies. HGPS mice were treated with tail vein injection of MTRT at around 3 to 6 months of age. Furthermore, treatments with this MTRT seemed to increase the lifespan of HGPS mice by 20%. In contrast, the untreated HGPS mice seemed to die off spontaneously. The researchers also identified that the earlier the treatment was administered, so like around at the 3-month mark, the more effective it was compared to like the 6-month mark. MTRT treatment also reduced blood clotting and induced a decrease of progerin in mouse cells as it did in human cells. This shows promise of using this treatment on cells inside living things. I made this line graph using the experimental data provided from the article to help demonstrate how the probability of survival increased for the MTRT treated mice versus their untreated counterparts. And as you can see, it is very clear that the treated mice were able to survive for several weeks longer than the untreated HGPS mice. Now, what about the limitations? So, no different to other great scientific breakthroughs and cutting-edge discoveries, there are obstacles that have hindered the experimenters' research. And I think it is really important to consider the limitations of this research to ensure an accurate understanding of our position in the advancement of this potential therapy. So the first significant limitation that caught my attention includes the HGPS differences between human patients and mice. For example, the HGPS mice did not develop occlusive atherosclerosis disease as do with the human children. Thus, we can make the inference that the physiology of the infected mice does not completely represent how the disease would progress inside humans. So therefore, it is still not certain how effective the treatment can be when conducting live human trials. Another crucial limitation is the delivery system. Due to the lack of targeting and modeling, the lifespan extension observed in the HGPS mice may be somewhat attributed to the non-vascular effects of MTRT treatment. Delivery of MTRT also has safety concerns because the genomic integration could potentially result in the immortality of cells, and that means it could also increase the capability of inducing tumors. However, despite these limitations, the experimental data suggests telomerase therapy as a viable solution for treating vascular disease in HGPS and potentially extending lifespan for these patients. Based on the promising results that I have presented from this study, the researchers have confirmed to be developing mRNA HTRT as a therapy for these affected children. And I believe it is very promising and could lead to great things, and this is just the beginning of this experimentation. Here are my own originally created diagrams that I used throughout the slideshow. So these ones I completely made all by myself, and several other diagrams throughout this presentation were simplified and customized by me as well. Here are the references and the websites I gathered information from. And thank you so much for listening to my presentation about telomerase therapy and its potential use and its promising future with helping HGPS patients. And I hope you're as excited as I am to see where this discovery takes us. Thank you.